Cancer is a disease that most of you have heard of, but some may not fully understand what it entails. In this lecture, we will explore the nature of cancer. Let's begin today's session. Topics to be covered in this lecture are: Number 1 is definition of cancer. Number 2 are types of cancer. Number 3 is growth and spread of cancer. Number 4 are causes of cancer. Number 5 is carcinogenesis, a multi-step process. Number 6 is treatment of cancer. Cancer. Cancer is a group of diseases characterized by uncontrolled or abnormal cell division. When cells in a part of the body divide without control, the excess tissue that develops is called a tumor or neoplasm. Neo means new. The study of tumors is called oncology. Onco means swelling or mass. Tumors may be cancerous and often fatal, or they may be harmless. A cancerous neoplasm is called a malignant tumor or malignancy. One property of most malignant tumors is their ability to undergo metastasis, the spread of cancerous cells to other parts of the body. A benign tumor is a neoplasm that does not metastasize. An example is a wart. Most benign tumors may be removed surgically if they interfere with normal body function or become disfiguring. Some benign tumors can be inoperable and perhaps fatal. Types of cancer. The name of a cancer is derived from the type of tissue in which it develops. Most human cancers are carcinomas. Carcin means cancer, omas means tumors, malignant tumors that arise from epithelial cells. Melanomas, melan means black, for example, are cancerous growths of melanocytes, skin epithelial cells that produce the pigment melanin. Sarcoma, sarc means flesh, is a general term for any cancer arising from muscle cells or connective tissues. For example, osteogenic sarcoma, osteo means bone, genic means origin, the most frequent type of childhood cancer, destroys normal bone tissue. Leukemia, leuc means white, emia means blood, is a cancer of blood-forming organs characterized by rapid growth of abnormal leukocytes, white blood cells. Lymphoma is a malignant disease of lymphatic tissue, for example, of lymph nodes. Growth and spread of cancer. Cells of malignant tumors duplicate rapidly and continuously. As malignant cells invade surrounding tissues, they often trigger angiogenesis, the growth of new networks of blood vessels. Proteins that stimulate angiogenesis in tumors are called tumor angiogenesis factors, TAFs. The formation of new blood vessels can occur either by overproduction of TAFs or by the lack of naturally occurring angiogenesis inhibitors. As the cancer grows, it begins to compete with normal tissues for space and nutrients. Eventually, the normal tissue decreases in size and dies. Some malignant cells may detach from the initial, primary, tumor and invade a body cavity or enter the blood or lymph, then circulate to and invade other body tissues, establishing secondary tumors. Malignant cells resist the anti-tumor defenses of the body. The pain associated with cancer develops when the tumor presses on nerves or blocks a passageway in an organ so that secretions build up pressure, or as a result of dying tissue or organs. Causes of cancer. Several factors may trigger a normal cell to lose control and become cancerous. Number 1 are carcinogens. Number 2 are oncogenes. Number 3 are oncogenic viruses. Number 1 carcinogens. One cause is environmental agents, substances in the air we breathe, the water we drink, and the food we eat. A chemical agent or radiation that produces cancer is called a carcinogen. Carcinogens induce mutations, permanent changes in the DNA base sequence of a gene. The World Health Organization estimates that carcinogens are associated with 60 to 90% of all human cancers. Examples of carcinogens are hydrocarbons found in cigarette tar, radon gas from the earth, and ultraviolet, UV, radiation in sunlight. Number 2 oncogenes. Intensive research efforts are directed towards studying cancer-causing genes, or oncogenes. When inappropriately activated, these genes have the ability to transform a normal cell into a cancerous cell. Most oncogenes derive from normal genes called proto-oncogenes that regulate growth and development. The proto-oncogene undergoes some change that causes it to be expressed inappropriately, to make its products in excessive amounts, or to make its products at the wrong time. Some oncogenes cause excessive production of growth factors, chemicals that stimulate cell growth. Others may trigger changes in a cell surface receptor, causing it to send signals as though it were being activated by a growth factor. As a result, the growth pattern of the cell becomes abnormal. Proto-oncogenes in every cell carry out normal cellular functions until a malignant change occurs. It appears that some proto-oncogenes are activated to oncogenes by mutations in which the DNA of the proto-oncogene is altered. Other proto-oncogenes are activated by a rearrangement of the chromosomes so that segments of DNA are exchanged. Rearrangement activates proto-oncogenes by placing them near genes that enhance their activity. Number 3. Oncogenic viruses. Some cancers have a viral origin. Viruses are tiny packages of nucleic acids, either RNA or DNA, that can reproduce only while inside the cells they infect. Some viruses, termed oncogenic viruses, cause cancer by stimulating abnormal proliferation of cells. For instance, the human papillomavirus, HPV, causes virtually all cervical cancers in women. The virus produces a protein that causes proteasomes to destroy p53, a protein that normally suppresses unregulated cell division. In the absence of this suppressor protein, cells proliferate without control. Some studies suggest that certain cancers may be linked to a cell having abnormal numbers of chromosomes. As a result, the cell could potentially have extra copies of oncogenes or too few copies of tumor suppressor genes, which in either case could lead to uncontrolled cell proliferation. There is also evidence suggesting that cancer may be caused by normal stem cells that develop into cancerous stem cells capable of forming malignant tumors. The process of inflammation, which is a defensive response to tissue damage. It appears that inflammation contributes to various steps in the development of cancer. Some evidence suggests that chronic inflammation stimulates the proliferation of mutated cells and enhances their survival, promotes angiogenesis, and contributes to invasion and metastasis of cancer cells. There is a clear relationship between certain chronic inflammatory conditions and the transformation of inflamed tissue into a malignant tissue. For example, chronic gastritis, inflammation of the stomach lining, and peptic ulcers may be a causative factor in 60 to 90% of stomach cancers. Chronic hepatitis, inflammation of the liver, and cirrhosis of the liver are believed to be responsible for about 80% of liver cancers. Colorectal cancer is 10 times more likely to occur in patients with chronic inflammatory diseases of the colon, such as ulcerative colitis and Crohn's disease. And the relationship between asbestosis and silicosis, two chronic lung inflammatory conditions, and lung cancer has long been recognized. Chronic inflammation is also an underlying contributor to rheumatoid arthritis, Alzheimer's disease, depression, schizophrenia, cardiovascular disease, and diabetes. Carcinogenesis, a multi-step process. Carcinogenesis is a multi-step process of cancer development in which as many as 10 distinct mutations may have to accumulate in a cell before it becomes cancerous. The progression of genetic changes leading to cancer is best understood for colon, colorectal cancer. Such cancers, as well as lung and breast cancer, take years or decades to develop. In colon cancer, the tumor begins as an area of increased cell proliferation that results from one mutation. This growth then progresses to abnormal, but non-cancerous, growths called adenomas. After 2 or 3 additional mutations, a mutation of the tumor suppressor gene p53 occurs and a carcinoma develops. The fact that so many mutations are needed for a cancer to develop indicates that cell growth is normally controlled with many sets of checks and balances. Thus, it is not surprising that a compromised immune system contributes significantly to carcinogenesis. Treatment of cancer. Many cancers are removed surgically. However, cancer that is widely distributed throughout the body or exists in organs with essential functions, such as the brain, which might be greatly harmed by surgery, may be treated with chemotherapy and radiation therapy instead. Sometimes surgery, chemotherapy, and radiation therapy are used in combination. Chemotherapy involves administering drugs that cause death of cancerous cells. Radiation therapy breaks chromosomes, thus blocking cell division. Because cancerous cells divide rapidly, they are more vulnerable to the destructive effects of chemotherapy and radiation therapy than are normal cells. Unfortunately for the patients, hair follicle cells, red bone marrow cells, and cells lining the gastrointestinal tract also are rapidly dividing. Hence, the side effects of chemotherapy and radiation therapy include hair loss due to death of hair follicle cells, vomiting and nausea due to death of cells lining the stomach and intestines, and susceptibility to infection due to slowed production of white blood cells in red bone marrow. Treating cancer is difficult because it is not a single disease and because the cells in a single tumor population rarely behave all in the same way. Although most cancers are thought to derive from a single abnormal cell, by the time a tumor reaches a clinically detectable size, it may contain a diverse population of abnormal cells. For example, some cancerous cells metastasize readily, and others do not. Some are sensitive to chemotherapy drugs and some are drug resistant. Because of differences in drug resistance, a single chemotherapeutic agent may destroy susceptible cells but permit resistant cells to proliferate. Another potential treatment for cancer that is currently under development is virotherapy, the use of viruses to kill cancer cells. The viruses employed in this strategy are designed so that they specifically target cancer cells without affecting the healthy cells of the body. For example, proteins, such as antibodies, that specifically bind to receptors found only in cancer cells are attached to viruses. Once inside the body, the viruses bind to cancer cells and then infect them. The cancer cells are eventually killed once the viruses cause cellular lysis. Researchers are also investigating the role of metastasis regulatory genes that control the ability of cancer cells to undergo metastasis. Scientists hope to develop therapeutic drugs that can manipulate these genes and, therefore, block metastasis of cancer cells. Thank you for watching. Like, share and subscribe my YouTube channel for further updates.